Human memory requires a physical substrate. To find it, we have to look closely at the dense, physical reality of the chemical synapse, specifically the microscopic gap separating the presynaptic vesicle pool from the postsynaptic density. A common misconception is that the strength of a connection between two neurons is fixed. In reality, synaptic efficacy is a highly dynamic variable, shifting constantly in response to neural activity. At the shortest timescales, ranging from a few milliseconds to a few minutes, we observe short-term synaptic plasticity. These are rapid, transient fluctuations in how effectively a synapse transmits a signal. This plot maps presynaptic action potentials on the top against the corresponding excitatory postsynaptic potentials, or EPSPs, on the bottom. Notice that when a second action potential arrives in rapid succession, the second EPSP peak is significantly larger than the first. This phenomenon is known as synaptic facilitation. The mechanism driving this facilitation is entirely presynaptic. When the first action potential fires, calcium ions flood the terminal to trigger transmitter release. If a second spike arrives before that calcium can be cleared, the residual calcium builds up, directly increasing the probability that subsequent vesicles will fuse with the membrane. This calcium-driven mechanism provides an immediate, efficient boost to the signal. But because calcium levels normalize rapidly, this short-term facilitation cannot serve as the biological hardware for storing long-term information. The transient nature of the synapse becomes even more obvious when we look at the converse phenomenon, short-term synaptic depression, which occurs when a neuron undergoes sustained high-frequency activity. This line graph tracks EPSP amplitude over time. When we apply a high-frequency sequence of stimuli, a tetanus, you can see a steep downward curve. The strength of the synaptic response rapidly drops off. This physiological depression is a simple matter of supply and demand. The intense stimulation exhausts the terminal's readily releasable pool of docked synaptic vesicles faster than they can be replenished. To restore the synapse to its baseline function, the neuron requires a brief refractory period to physically mobilize a new supply of vesicles to the active zone. These rapid presynaptic fluctuations are useful for immediate, localized computational tasks in the brain. But human memory, the storage of faces, routes, and experiences, must endure for months or years without degrading. Because presynaptic resources inevitably deplete under heavy load, the brain must rely on entirely different, postsynaptic mechanisms to achieve permanent structural memory. To find those mechanisms, we look to the mammalian hippocampus, the primary biological structure responsible for memory formation and spatial navigation. This schematic maps the complex trisynaptic circuit of the hippocampus. The critical pathway for our purposes is the projection of the Schaffer collateral axons, which travel from the CA3 pyramidal neurons directly to the dendrites of the CA1 pyramidal neurons. It is at this specific junction that we can consistently observe long-term potentiation, or LTP, a persistent, long-lasting increase in synaptic efficacy that follows a brief burst of high-frequency stimulation. This graph illustrates the baseline LTP experiment. It tracks two distinct datasets over 60 minutes. The control pathway, receiving normal stimulation, remains flat, but the tetanized pathway, the one exposed to a high-frequency burst, shows a sustained, elevated EPSP amplitude that does not return to baseline. If we extend this timeline to match in vivo observations, compressing the x-axis out to a full year, those data points remain elevated without degradation. This extraordinary temporal persistence proves that the CA1 synapse possesses the necessary biophysical stability to act as a lifelong matrix for memory. But LTP does not happen arbitrarily. For the brain to commit resources to a memory, the neural network requires strict computational rules. The first is coincidence detection. A weak, isolated release of presynaptic neurotransmitters is entirely insufficient to trigger long-term changes in the postsynaptic receiving cell. This split timeline demonstrates the precise threshold condition. On the left, weak stimuli yield no permanent change, but on the right, when those exact same weak presynaptic action potentials are artificially paired with a strong depolarization of the postsynaptic membrane, LTP is immediately induced. This specific requirement was predicted in 1949 by Donald Hebb. Hebb's postulate states that cellular learning requires tightly linked, simultaneous activity in both the transmitting and the receiving neurons. This strict dual activation requirement acts as a biological filter. It ensures that the network only expends metabolic energy to store highly correlated, meaningful data, safely ignoring random baseline electrical noise. To store vast amounts of discrete information without interference, the network relies on a property known as input specificity. When LTP is induced at one highly active synapse, it physically strengthens. But critically, the adjacent inactive synapses located on the exact same dendrite remain completely unaffected. However, this localized specificity is balanced by a complementary network rule, associativity. If that adjacent, weaker synapse fires at the exact same moment the strong synapse is active, both connections strengthen simultaneously. Through associativity, a weak input can essentially hitchhike on the strong one. Because the strong input successfully depolarizes the shared postsynaptic cell, the coincident weak input undergoes LTP. These precise properties provide the required cellular hardware for classical conditioning. They allow the brain to physically link previously unrelated environmental stimuli, mapping complex associations directly into the architecture of the dendrites. We can trace this Hebbian logic down to a specific molecular architecture located precisely at the postsynaptic density. The physical embodiment of the brain's coincidence detector is the NMDA receptor. This cross-section of the NMDA receptor displays its dual activation biophysics. The channel only opens when two distinct events happen simultaneously. Presynaptic glutamate binds to the receptor, and postsynaptic depolarization physically repels the resident magnesium ion blocking the pore. Once that block clears, a massive influx of calcium ions floods into the cell. This sudden calcium surge triggers the early phase of LTP. The ions bind to and immediately activate local intracellular kinase complexes stationed just inside the postsynaptic membrane. The immediate structural result is the rapid trafficking and insertion of additional AMPA receptors directly into the membrane. This thickens the active zone, permanently increasing the cell's sensitivity to future bursts of glutamate. This rapid receptor upgrade provides an immediate, functional boost to synaptic gain. It secures the memory trace in the short term, entirely bypassing the slower process of cellular gene transcription. Adding surface receptors will secure a memory for hours or days, but to sustain a memory for a lifetime, the neuron must initiate the late phase of LTP, a process altering the cell's actual DNA expression. This macro view tracks the late phase signaling cascade. Intracellular enzymes, specifically protein kinase A, travel from the activated dendritic spine down the dendrite and directly into the cell nucleus. There, they activate a transcriptional regulator called CREB, triggering mRNA transcription. Once activated, this gene expression initiates the large-scale synthesis of entirely new synapse growth proteins. This microscopy image captures the physical evidence of that synthesis. You can clearly see multiple glowing, newly formed dendritic spines budding directly off a mature CA1 dendrite to form novel synaptic connections. True long-term memory is architectural. To house permanent information, the brain literally builds new physical hardware. This powerful capacity for structural change comes with severe clinical risks. If the mechanisms of synaptic plasticity are left unregulated, the network degrades. We see this acutely in epilepsy, a primary pathology of abnormal synaptic plasticity, where the network falls into a state of intense, pathological synchronization. This electroencephalogram traces the electrical signature of a seizure. Notice the transition from chaotic, independent healthy wave patterns into massive, synchronized, high-amplitude pathological spikes. These intense, synchronized bursts act as artificial tetanic stimuli, incorrectly triggering LTP mechanisms across the cortex. The brain carves pathological pathways that strengthen the abnormal connections, making future seizures more frequent and more severe. Synaptic plasticity exists as a delicate, high-stakes balance between rigid neural stability and chaotic overconnection. From the millisecond dynamics of calcium fluxes in the presynaptic terminal to the permanent, year-long architectural remodeling of dendrites, The biophysics of the synapse serve as the literal physical substrate of all human learning, memory, and identity.